Hello class, today I'm going to talk with you guys about protein synthesis. Protein synthesis is the process by which our DNA is transformed into us, flesh and blood. So this graphic here, this image here probably looks very confusing, but hopefully by the end of this screencast you'll be able to understand exactly what's going on in this image. So, the central dogma of molecular biology is the process by which DNA is transcribed into RNA, and then RNA is translated into a protein. Now remember, previously we learned the difference between DNA and RNA, and now you're going to be able to fully understand the role of DNA in protein synthesis and why DNA is so important. So, anytime a cell begins to grow, it has to copy its DNA so that it can have an extra duplicate copy for the new cell that it's preparing. So DNA replication is just the process by which a strand of DNA is replicated or a copy is made. So it's really important during that process that no mistakes are made. The cell is very, very miraculous in that it has a whole entire process that allows replication to occur, and in order for replication to occur, the DNA has to be transcribed and translated, and new proteins have to be made. So let's start with the process of transcription. First question that you should ask yourself is where does transcription occur? And transcription occurs inside of the nucleus because that is where all of your DNA is stored. And when I tell kids the best way to memorize transcription, the word script means to write down something or to scribe. And so the process of transcription is to write down or copy the DNA sequence. Because the DNA is double-stranded, it is too large to actually leave the nucleus through the nuclear envelope. So because it cannot leave, the DNA remains inside the nucleus. The job of the mRNA is to take one of those strands of DNA and actually copy down the complementary sequence. We know that DNA contains the nucleotide bases adenine, thymine, guanine, and cytosine. And so, based off of those sequences, the letter sequences, mRNA strand can be produced. Now, mRNA is actually a single strand. And because it's single strand, it can actually escape through the pores of the nucleus and go out to the ribosomes where proteins are actually made. Before the mRNA leaves the nucleus, the mRNA goes through this processing. And in this processing, it gets rid of chunks of DNA that don't really particularly code for anything, and it prepares the mRNA to be translated into an actual protein. The most important thing for you to understand in that transcription process is that the DNA is copied into RNA, and then the RNA is then allowed to leave the nucleus in order to go to the cytoplasm and to go to the ribosomes, in which the location of the proteins are actually produced. So there's more details involved. If you look closely at this graphic, kind of gives you an idea that when you're looking at the blue strand here, the blue strand represents the actual DNA. So there's two sides to a DNA strand. Notice it's the letter sequences, the A for adenine, the T for thymine, cytosine, and so forth. The job of the mRNA is to take one of those strands and make a complementary base for that strand. So if the DNA is T, then the RNA is going to be A. If the DNA is A, then the RNA, instead of being T, remember there's no thymine in RNA, it becomes U for uracil. That's probably one of the biggest things that's difficult for most kids to remember, is that there's no T in RNA. The T becomes a U for uracil. The G and C still bind together, so wherever you have a G, it becomes a C. Now, and remembering also in all of that, this base pairing process is pretty much done from the 5' to the 3' on the DNA molecule. Once that RNA copy is made, it is then taken and released to go into the nucleus. Go into the ribosomes. Ribosomes is the location of translation. So transcription just means you're scribing or you're scripting or writing out the actual letter sequences of the code, that DNA code, which is your blueprint. When you go into the process of translation, I always tell my students to think of translation as if you're reading a foreign language. If you're trying to translate from English to Spanish or Spanish to English, what does the letters or the words sound like or mean? So those letter sequences, that A, that T, that C, and that G, are transcribed into the RNA, which then goes to the ribosomes. And they act like little recipes. And these little recipes, the ribosomes take them and they transform them into proteins. So the translation process, if you can think about it in terms of translating it into the language of life or coding for flesh and blood. So transcription, DNA to RNA, translation, that RNA now becomes the actual protein. And all of this happens in the cytoplasm with the ribosomes. Now we're going to introduce a new player. We talked about the 3 types of RNA. So mRNA is the messenger RNA. It's the one that has the actual message or code copied. But then the tRNA is the transfer RNA. And this RNA has a special job of transferring amino acids. So when you look at the information in the mRNA, every 3 letters represents a codon. A codon is one word in the recipe. So the mRNA is read as every 3 letters codes for one amino acid. Amino acids are the building blocks of proteins. Your genetic code just is a list of letters that translates into a series of amino acids that eventually will link together and become proteins. This chart is the amino acid chart. There are actually 20 different amino acids, and there are 4 different nucleotide sequences. So when you're doing your mRNA, you have to figure out the 3-letter code to represent which amino acid. So I'm going to do a couple of examples with you. You will get pretty good at it once you practice it for a little bit. The inner circle represents the first nucleotide base. The second circle represents the second base. And the third circle, the outermost circle, represents the third nucleotide base in your 3-letter codon. So let's say I have a DNA sequence that reads TCG, thymine, cytosine, guanine. Well, my RNA sequence will transcribe that into A, G, C. In order to figure out what my amino acid is, I have to start from the center. In order to do that, I start with the A, that's the first sequence. I go out to the G, that's the second sequence. And then I go to the C, which is the third. The amino acid for that sequence would be serine. And then that's how it knows which amino acid codes for which codon in the sequence chain. Now let's work through another example. Let's see if you can do this one. I'll give you a minute. If my original DNA is GAG, first you have to transcribe it into RNA. The RNA sequence, if you got it right, would be CUC. Remember, there's no T in RNA, so that A would become a U. And then what would be the amino acid sequence that would go along with the RNA from this codon chart? If you go from C to U to C, you would get leucine. So this process of translating involves using this codon chart to see what every 3-letter code represents. I wanted to show you this chart as well. You'll see it written both ways. The other chart I think sometimes is easier for some kids, but some kids like this chart instead. Same concept. If I say my DNA is AAA, then I know my RNA is going to be UUU, and that translates to phenylalanine. So it's another form that you'll see it in. But either way, if you see it in the circular chart or if you see it in this more traditional chart, you should be able to know that this is the first base, and the red over here, and then the top is the second base, and then the third base is in the green, and it still codes for the amino acid that it needs to. So when you're looking at translation, here's more of I like a visual of it. Keeping in mind, if this is the nucleus, the mRNA leaves the nucleus, it goes to the ribosomes, and once it's at the ribosome, it begins to read each three-letter codon, and as it reads each three-letter codon, these that are floating in the cytoplasms are the transfer RNA. Notice the transfer RNAs have amino acids attached to them. So the transfer RNAs have what we call the anti-codon attached to them. So when you look a little closely, if this is the tRNA molecule, the codon is CUA, well, the tRNA molecule will have the anticodon linked to it. That's how they know exactly how to match it up. And then the anticodon is going to be a reflection or the complementary of the mRNA. And when it matches up, then it releases that particular amino acid. Once those amino acids are released, they are linked together by peptide bonds to form what we call the polypeptide, which then eventually will fold and become the new amino acid. This process will always start with a start codon, and it will end with a stop. So once it gets to the stop codon, that amino acid will stop recording and coding, and that polypeptide chain will fold up and become your new protein. So transcription, translation. Anytime you get to a stop codon, it immediately breaks it off. The start codon is always going to be AUG. AUG corresponds to methionine or MET. This is the initial or the first amino acid. It will continue to code every three amino acids until it gets to the stop. Once it gets to a stop codon, like UAA, it'll tell the process to stop there. And then that will become your polypeptide. So, going back to the original picture, from gene to protein, if I look at this picture and I ask you, where does transcription occur, you should be able to tell me in the nucleus. And if I ask you, why does it occur in the nucleus, you should be able to say that the DNA molecule is too large, so the RNA has to make a copy of it. If I ask you the role of the mRNA, you should be able to say transcription, or copying down the genetic code. When we talk about translation, if I say, where does translation occur, you should be able to say it happens at the ribosomes and also in the cytoplasm. And if I ask you what the role of the tRNA molecule is in protein synthesis, you should say they actually carry the free amino acids that are floating in the cytoplasm to the ribosomes, where they can be attached to make the actual polypeptide or protein. And I hope that this really helps you understand the process of going from a gene to a protein.